Welcome to episode 38 of the Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we're exploring cardiac muscle cells, the specialized muscle fibers that keep your heart beating rhythmically and efficiently. Now, unlike skeletal muscle, cardiac muscle has unique properties that allow it to contract continuously without fatigue. In our next episode, we'll build off of this and focus on stroke volume. But for now, let's break down the action potential of cardiac muscle cells, the role of calcium in contraction, and how cardiac muscle differs from skeletal muscle. Before diving into the action potential of cardiac muscle cells, let's compare it with skeletal muscle. Now, cardiac muscle cells, with the help of cardiac pacemaker cells, are autorhythmic, meaning they can generate their own action potentials without input from the central nervous system. This is different than skeletal muscle, which requires that nervous system input. Intercalated discs and gap junctions allow cardiac cells to contract as a unit. We call this a functional syncytium, while skeletal muscle fibers contract independently. Cardiac muscle cells have a plateau phase in their action potential, preventing tetanus and allowing for proper relaxation between beats. Now, a contractile cardiac muscle cell action potential occurs in 5 distinct phases. There's depolarization. Sodium influx through voltage-gated sodium channels rapidly depolarizes the membrane. This phase is similar to skeletal muscle depolarization, but leads to a longer action potential. Next, we have early repolarization. During this phase, some potassium channels open, and these are also voltage-gated, which causes a small repolarization. This phase ultimately helps prepare the skeletal muscle cell for the plateau phase. Now, during the plateau phase, calcium influx through voltage-gated calcium channels balances out that potassium efflux, which creates a sustained depolarization. This phase lengthens the action potential, ensuring proper contraction and relaxation cycles. The plateau prevents tetanic contractions, which would be fatal for the heart since it needs to fully relax between beats. At the end of the plateau phase, we have the true repolarization phase. This occurs when calcium channels close while potassium efflux continues, ultimately restoring the resting membrane potential, and this allows the muscle to reset for the next contraction. And so we're back to resting membrane potential, where the cell returns to its resting state, waiting for the next signal that causes depolarization. Now, the plateau phase is unique to cardiac muscle cells and serves several important functions. First, it prevents tetanus or a sustained contraction, which ensures rhythmic heartbeats. Additionally, it allows sufficient time for the ventricles to fill before the next contraction. And finally, it prolongs the refractory period, which prevents premature contractions that could disrupt cardiac function. Now, like skeletal muscle, cardiac muscle contracts via the sliding filament mechanism, but calcium regulation is slightly different, as calcium is released through 2 pathways. First, we have the ryanodine receptor activation, which is similar to that found in skeletal muscle. The action potential activates voltage-gated calcium channels, which triggers calcium release from the sarcoplasmic reticulum. More calcium binds to troponin, which exposes binding sites for myosin on actin, allowing for greater contraction. Remember, more calcium release equals a stronger contraction. That's the major pathway. Then we have the minor pathway of IP3. The G protein pathway activates phospholipase C, which converts PIP2 into DAG and IP3. IP3 stimulates additional calcium release from the sarcoplasmic reticulum, contributing to contraction. This pathway is less significant in cardiac muscle, but can help fine-tune contractility. Now, if we want to, we can compare action potentials between cardiac pacemaker, cardiac muscle, and skeletal muscle cells. Pacemaker cells self-generate action potentials to initiate heart rate. Cardiac muscle cells contract in response to cardiac pacemaker cell activity, which helps maintain that rhythmic heart rate. And then there are skeletal muscle cells. These cells require external nerve stimulation and can undergo tetanic or sustained contractions. So let's summarize with our 4 key takeaways. Cardiac muscle action potentials are longer than those of the skeletal muscle cells, and this is because of the plateau phase. Calcium influx during the plateau phase is essential for proper contraction and preventing tetanus. Ryanodine receptors play a major role in calcium release, while the IP3 pathway has a minor influence. Cardiac muscle cells contract as a unit thanks to intercalated discs and gap junctions. And so that's it for episode 38 of the Pulse of Physiology. I hope this episode has given you a deeper understanding of cardiac muscle cells and their role in maintaining heart function. Join me next time for episode 39, where we'll discuss stroke volume and how the heart adjusts force generation to meet the body's demands. Until then, keep questioning, keep exploring, and stay curious.